Hi everyone, it's Mrs. Bustamante. Today we're going to talk about the structure and function of those biological molecules we've been spending so much time on. But before we really get into that, I want to talk about something called directionality. So how these biological molecules are put together or built matters. They actually have a three-dimensional structure to them, and we kind of, I think, forget that sometimes because we're looking at them on a piece of paper or on a flat page or on this presentation, right? So what do I mean by directionality? Well, it kind of matters how they're put together and how we look at them. Think about our hands, right? They look the same, but I can't put one hand on top of the other and have them match up because my hands have directionality. It matters how they're placed together. So these are actually 3D objects, and we have to remember that when we're looking at how they're placed together. Let's go ahead and start by talking about carbohydrates. Remember, carbohydrates are chains of sugars, and they can either be what we call straight or they can be branched. And this is really important in determining the function of what that carbohydrate will do, right? We've talked a lot about structure determines function. You've heard that over and over and over again. So really how they're put together or structured will determine the function of that carbohydrate. Here, for example, we have amylose and we have amylopectin, and they are both polymers of glucose, the sugar glucose, but you'll notice that they're put together differently, and so they have different functions. Amylopectin actually is used in like fruit jellies to kind of help it stay gooey, I guess you could kind of say. So carbohydrates, what's their goal? Well, they store energy, but they kind of are a quick release energy. It doesn't take as much energy to build them, so we can give off their energy really quickly. Things we think about: carbohydrates, starch, glycogen, which is found in liver and muscle cells, which often need energy, right? So good things. Carbohydrates tend to give us quick energy, if you will. One that I always think is interesting to talk about is starch versus cellulose. Okay, and starch and cellulose are both made of carbohydrates, but they're isomers of each other. So what does that mean? Well, it means they have the same chemical formula, but how those molecules are put together, their structure of them is actually a little bit different. So they're gonna have different functions, right? They do different things. And what we notice about this is starch, we see our glycosidic bond, right, the bond that holds sugars together, is all on the same side of our glucose molecules. Another way to say that is it lies flat. Cellulose, on the other hand, you'll notice that the glycosidic linkage, right, kind of alternates up and down, or it's a cross linkage. Cellulose is found in plants a lot, and it's really hard for lots of mammals to digest because of this. However, cellulose happens to be the most abundant organic compound on Earth. And if we think about that, it's because there's lots of herbivores, it's in plants, and we need plants to support kind of everything else, right? So this cross-linking between these polysaccharide chains means that it's rigid, it's hard to digest for a lot of organisms, which is different than starch. So what does that mean? When we think about herbivores, they're kind of specially made, if you will, to be able to digest cellulose very well. Where if you're not just an herbivore, it's a lot harder for you to digest cellulose because of this specific linkage. And because if you're not an herbivore, say like a cow, for example, that's basically made to digest cellulose in a really slow fashion without a lot of microbes present, it's harder for you. You have to eat other foods and other simple sugars and provide other nutrients in order to get what your body needs. So I think that's a neat example of starch and cellulose and being both made of glucose. Next, let's go ahead and talk about proteins. So if we go back to proteins, remember they have an amino group, they have a carboxyl group, they have this hydrogen, and they always have this carbon at the center of them. And then they have this R group that can change. Sometimes we call the amino group the N terminus, and the carboxyl group the C terminus. And new amino acids, when we build them, are always going to add to our carboxyl group. And they're going to add in a linear fashion. And what are some functions of proteins? Lots of enzymes, transport, signals, defense, structures, receptors, all very important. And when we look at this one, right, we see our C-N chain, and then we have our R groups down here that can have a lot of variance. And when we think about our R groups, remember they can be hydrophobic, hydrophilic, they can be charged, they have a lot of different properties. They're kind of where the uniqueness will come about. So proteins, if we take it a step further, they have different levels of structure. So the primary structure of proteins, or the first level of protein structure, is basically just the amino acid sequence in the polypeptide chain. It's determined from your DNA sequence, right? You look at your DNA code and that will code for a specific amino acid, and then we just put them together. So a slight change in your protein structure here can really affect your function, right? And sometimes even if it works or if it doesn't work how it's supposed to. So primary structure is really just your amino acid sequence. Secondary structure of proteins is how these proteins fold together based on their primary structure. So they can fold along short little sections of the polypeptide chain and basically with amino acids that are close to them. This is typically based on hydrogen or weak bonds in the R groups. There are two main ways that they fold that we should know about. One of them is the alpha helix. It looks kind of like a spiral. And one of them is the beta pleated sheets. And it looks kind of like if you took a piece of paper and you folded it into a fan, what it would look like. Proteins can have both of these in a protein itself, or just one of them. It can really vary. So that's your secondary structure. Your tertiary structure is then the 3D folding of this protein, and it's really determined by your R groups. Remember, your R groups are the parts, they can be hydrophobic, they can be hydrophilic, and they can be charged. And it's the interactions of these R groups that determines this 3D structure of how these proteins fold, based on hydrogen bonding, ionic bonding, and something called disulfide bridges, where the sulfur in them will bond with hydrogen. Hydrophobic R groups tend to fold towards the inside of a protein because if we think about where these proteins are, they're in the cytoplasm, and cytoplasm is like water-based, so it's polar, right? So they're going to move away from the polar outside because they're hydrophobic. And then hydrophilic R groups are going to tend to be on the outside of the protein and be surrounded by the cytoplasm. All proteins have three levels or three structures. They all have primary, secondary, and tertiary structure. Only really complex proteins have what we call quaternary structure, which is the fourth level. And this happens when we have multiple polypeptide chains that interact and interact together. So a good example of this is hemoglobin. Hemoglobin is actually built on 4 polypeptide chains, and so how they form together. And again, we are going to see parts of it on the outside near the cytoplasm and parts on the inside based on hydrophilic interactions. So something else that's interesting about proteins, we can denature proteins. They have to be kind of at a very specific pH and a very specific temperature, and even a salt concentration or charged ions can vary it because it can break up the interactions in those proteins. And again, structure determines function, which oftentimes denaturing a protein will mean it doesn't function the way it should. So that's a really important part about proteins. They kind of have to have the ideal environment. So this is a nice picture to walk you through. The primary structure is just the amino acid sequence. Secondary structure is the folding of the polypeptide chain into beta sheets or alpha helixes. Tertiary structure is the 3D folding of those based on the R groups. And then if you have multiple polypeptide chains, that's your quaternary structure, how they interact together. Up next, we have nucleotides. Nucleotides have a phosphate group, and it's always going to be at what we call the 5' end or the 5' carbon, and they have a hydroxyl group, and it's always going to be attached to what we call the 3' end. Why is this 5' and 3' end really important? Well, it's really important for DNA and RNA synthesis because new nucleotides are always going to be added at this 3' end. They cannot be added at the 5' end. And they form a covalent phosphodiester bond when they do this. So this kind of plays into an important role about DNA. DNA, we say, is anti-parallel. It's two anti-parallel strands. We notice that the 5' end of one side of the DNA molecule matches with the 3' end of the other side of the DNA molecule. So they are anti-parallel. They are opposite in the direction of each other. So you'll notice that adenine will always form or bond with thymine, and it will form two hydrogen bonds between those two molecules. And cytosine will form with guanine or bond with guanine with 3 hydrogen bonds. So that will become important a little bit later on when we talk about how DNA replicates and what those hydrogen bonds mean. But that is important to know. And remember that we don't have thymine in DNA, or excuse me, in RNA. In RNA, we have uracil. So uracil will replace thymine. Lastly, we have our lipids. Lipids, remember, they're really big molecules. They have these huge hydrocarbon chains, and carbon-carbon bonds tend to hold a lot of energy in them. So lipids tend to have be used for long-term energy storage. They tend to be concentrated with those hydrogen and carbon bonds, and there's lots of them. And we think big energy because they're big molecules. So sometimes we can think about why do we desire fats, right, as humans? Well, if we think back to a long time ago when people lived on like the Serengeti Plain and didn't have a lot of resources, right, as far as food goes, fats would be important because you could use that energy for a long amount of time and store that energy to use for extended periods of time versus something that might just give you quick energy. I hope that was helpful, and we'll talk soon.